Welcome to Biology at Priyojit, my dear viewers. Today we'll dive deeper into enzyme action to understand how enzymes actually work, what factors influence their activity, how inhibitor molecules regulate enzymatic reactions or enzyme catalyzed reactions, and why allosteric enzymes play a crucial role in metabolic control. From temperature and pH to competitive and non-competitive inhibition, and from active sites to regulatory sites, these mechanisms finely tune biochemical reactions inside the cell. So let us explore these concepts in a simple, concept-based and exam-oriented manner so that enzyme regulation becomes clear, logical and easy to remember for university and competitive exams. Factors affecting enzyme action. The rate of an enzyme catalyzed reaction is influenced by the following factors. Temperature. Enzyme activity increases with rise in temperature but up to a certain limit. The image showing the x-axis represents the temperature and y-axis represents the rate of reaction. So up to a certain limit, when temperature is increases, rate of enzymatic reaction is also increases, but it occurs up to a certain limit. After that limit it falls down. Beyond this limit enzyme denature, and so it lost its structure and activity also decreases. So what is this temperature is optimum temperature, that is the temperature at which enzyme activity is maximum. That temperature is called as optimum temperature and it is specific for a particular enzyme. Basically, most human enzymes have optimum temperature between 37 to 40 degrees Celsius. At 0 to 4 degrees Celsius, we know enzymes are almost inactive. So this is the basis for food preservation in refrigerator. During fever, our increased body temperature also increases enzymatic activity and it increases the metabolism also. So what is hypothermia? Reduced enzyme activity, which is the concept is used in case of organ transplantation, open heart surgery, etc. So enzymes in extremophiles, hot spring bacteria, they are very high optimum temperature. Cold adapted bacteria, their optimum temperature is nearly about 4 degrees Celsius, but there is stack polymerase which acts on optimum at 42 degrees Celsius. Now, according to scientist Van 't Hoff, there is a concept Van 't Hoff's coefficient or Q10. According to this concept, for every 10 degree Celsius rise, enzyme activity doubles till its optimum temperature. pH. Each enzyme has a specific optimum pH and enzyme activity decreases if pH is above or below the optimum level. So basically most enzymes work best at pH 7.4. So examples, there is pepsin pH 1.5, acid phosphatase pH 4.5, alkaline phosphatase that is pH 9.8. So we know what is the conclusion. Like temperature, each enzyme has a specific optimum pH, and when pH is increases, the rate of enzyme catalytic reaction will be also increase and up to certain point, and that particular pH enzyme activity decreases. So if higher or lower H plus ion in acid or OH medium in alkaline can interfere with enzyme structure. Most examples such as pepsin acts at pH 1.5, acid phosphatase acts at pH 4.5, alkaline phosphatase acts at pH 9.8. Substrate concentration, increasing substrate concentration increases the reaction rate of an enzyme catalyzed reaction. At high substrate concentration, what we know, that enzyme becomes saturated, and after saturation rate becomes constant. So the reaction will achieve or reach at the Vmax, that is the maximum velocity. So graph of the velocity versus substrate concentration, it gives a hyperabolic, rectangular hyperbolic graph. Increased substrate concentration causes increased rate of reaction until the active site of the enzyme are used. Enzyme concentration with excess substrate. Increase in enzyme concentration also increases reaction rate, and relationship is directly proportional like a straight line, and here the graph obtained is a straight line graph, rate of reaction and enzyme concentration, and this in the graph we can find until substrate amount are limited. Product concentration, we know that accumulation of product can slow down the enzyme activity. This occurs due to feedback inhibition. Light, certain wavelength affect enzyme activity. Say for example, we can find it in kind of photosynthesis. We know that in blue light increases activity of salivary amylase. Salivary amylase activity increases in blue light, and UV light decreases its enzymatic activity. So light is an important factor for controlling enzyme activity. Ions, some enzymes require specific ions for its activity, such as pepsinogen requires H plus ions, kinases require Mg2 plus or magnesium 2 plus ions. Now we'll go for units of enzyme activity. Enzyme unit or IU, that is the amount of enzyme that converts 1 micromole of substrate per minute at 25 degrees Celsius. Specific activity, enzyme units per milligram of protein is denoted by enzyme specific activity. It indicates enzyme purity. What is Catal or what is the SI unit? Amount of enzyme that converts 1 mole of substrate per second is called as Catal, or it is the SI unit, and 1 Catal is equivalent to 1 mole per second. So what is turnover number? We know that number of substrate molecules converted per unit time by one specific enzyme molecule is that the turnover number of that particular enzyme. So we know that carbonic anhydrase has highest turnover number, that is 36 million per minute. That means in a single minute carbonic anhydrase enzyme can convert 36 million molecules into product. Enzyme inhibition. Enzyme inhibitors reduce enzyme activity. Type of inhibitor: irreversible inhibitor and reversible inhibitor. Irreversible inhibitor bind covalently to enzyme. It causes permanent inactivation of the enzyme. It modify essential amino acids like serine, cysteine, threonine and tyrosine. It cannot be removed by dilution or dialysis. Reversible inhibitors, these reversible inhibitors bind non-covalently with the enzyme, and this binding can be reversed. So there are basically three types of reversible inhibition: competitive inhibitor, non-competitive inhibitor and uncompetitive inhibitor. So we can conclude that enzyme inhibition can be of two types: reversible and irreversible. And reversible inhibition can be of three types: competitive, uncompetitive and non-competitive. So we have already discussed the differences between reversible inhibitors and irreversible inhibitors. So reversible inhibitors, we know that they bind non-covalently. The binding can be reversed, and there are three types: competitive inhibition. Here inhibitor molecule competes with the substrate for that particular active site, and this competitive inhibition can be overcome by increasing substrate concentration, and inhibitor resembles the substrate structurally. Uncompetitive inhibition, here inhibitor binds to enzyme substrate complex. It cannot be overcome by substrate increase. Binding alters enzyme conformation. Non-competitive inhibition, here inhibitor binds at allosteric site and changes shape of active site, and substrate concentration has no effect in the process. So this is reversible enzyme inhibition: competitive inhibition, non-competitive inhibition and uncompetitive inhibition. Here enzyme inhibition, competitive inhibitor and non-competitive inhibitor, which depends on substrate concentration. Allosteric enzymes and their kinetics. Allosteric enzymes basically have quaternary structure, that is it has multiple subunits like a protein, and so it has possessive active site, possesses active site and allosteric site or regulatory site or controlling site, and this allosteric enzyme can be exist in T state or 10 state and R or relaxed form. So they show sigmoid or S-shaped curve due to cooperative binding, and they do not follow Michaelis-Menten kinetics. So modulators are the molecules that can bind to allosteric site and that can be either activators or inhibitors. Homotropic enzyme, when substrate itself acts as modulator, they're called homotropic enzyme, and when modulator is different from the substrate, that is called as heterotrophic enzyme. So this is the image showing the allosteric inhibition and allosteric activation. This is the allosteric site where an inhibitor can bind, and this is the allosteric site where an activator can bind. Models of allosteric enzyme action. There are two models. Sequential model proposed by Khosla in 1966. So binding of substrate to one subunit induces change only in that subunit, and symmetry is not maintained in this according to this model. And concerted or symmetry model, according to Monod 1965, all subunits change together and the activators bind to the relaxed form, that is there is two form, relaxed state and tense state, and inhibitors bind to the tense form. So here symmetry is maintained overall. So what is catalytic versus regulatory enzymes? We know that regulatory enzymes control metabolic pathways, usually the first enzyme in a pathway, and regulation saves energy and resources. Regulation occurs by allosteric modulation and covalent modification like phosphorylation, methylation, ADP ribosylation, etc. So examples of these catalytic versus regulatory enzymes include chemotaxis protein, which is regulated by methylation, and ADP ribosylation also uses NAD as a donor. So my dear students, we have seen that enzyme activity is finely controlled inside living cells, finely regulated inside living cells. Factors like substrate concentration, temperature, pH and enzyme concentration influence the rate of enzyme catalyzed reactions. And enzyme inhibition helps control metabolic pathways, while allosteric enzymes play a key role in precise regulation through regulatory sites. Together, these mechanisms ensure efficient and controlled metabolism, making enzyme regulation a vital concept in biochemistry. So keep revising, stay curious, and let enzymes reveal the chemistry of life to you. Thank you, and see you in the next class.